So, great way to start out the week. We're delighted to welcome Professor Karl Deisseroth from the Departments of Bioengineering and Psychiatry and Behavioral Sciences at Stanford. Karl did his undergraduate work at Harvard and then his MD and PhD at Stanford. He's also a practicing psychiatrist, maybe the first one to talk from this podium. His combined expertise in life sciences and engineering fits really well with the vision of Technion and of our department and how life sciences fits into Technion. So we're really especially pleased and congratulate you on receiving the Harvey Prize today. Professor Deisseroth is a Howard Hughes Medical Institute investigator and member of the National Academy of Sciences. And to keep the introduction reasonably short, I'll just mention that this is not the first of his big awards. Karl's best-known contribution is, of course, the development of optogenetics. As a photobiologist, I'm honored to give this introduction because even as a young student, I was fascinated by light as a signal. As Hartmann said and wrote in 1972, photons can be applied with light velocity or potentially without inertia, regardless of whether the on or off problem is considered. So lack of inertia means that you can touch the biochemistry of a cell without touching the cell. You don't have to put in any molecule. So just you shine light and things happen. And I really don't know what the on or off problem meant back then. But in the context of optogenetics, it's certainly the ability to switch neuronal activity on or off using light absorbed by engineered ion channels. And this has opened up an endless world of possibilities to control and understand the function of the brain. The Deisseroth Lab has gone on to do other things, other fascinating connections between optical technology and structure and function in the brain, and we're really looking forward to hear about that today.
All right, thank you. It's an honor to be here, to come back to Israel, my first time in Haifa. I had a nice tour last night, so thanks to the wonderful hosts who took me around, and I'm looking forward to sharing our work, including a lot of unpublished work today, but I'll also, in the context of the prize, give a little basic background on optogenetics and things we've done in the past. But I'll get as quickly as I can to the new work. I think everybody here probably appreciates the beauty of the brain and its complexity. This is a zebrafish brain where you can see activity of individual neurons flashing. I'll come back to this sort of data from my lab later. But with all that complexity of activity, to understand it, one has to overlay onto that understanding of connectivity and wiring, and we have strategies for doing that as well. How are those active cells wired up? And then finally, it's not, of course, enough to observe what happens. As biologists, one wants to test perturbations, and to do so in a setting that is of interest, for example, in behavior. And in behaviors that are of interest to us, here's an animal that is spending time in one part of a maze and not another, and I'll get back to the reasons for that shortly. But one thing we're trying to do is bring all these different data streams together in the same preparation at the same time, so we understand the natural activity, the causal activity, the impact on behavior, and the wiring, and try to understand integrative principles altogether. The optogenetic method, as you all know, confers on us a capability that electrical and magnetic interventions do not. Just like in the beautiful introduction, and we'll have to get that quote from you, the inertia quote. I think that's a very perceptive one. I hadn't heard that before. But there are the other advantages of light in this context, not just the lack of inertia, but also the targetability. This directly addresses this challenge that since all neurons are electrical, and electrical or magnetic to electrical intervention can't distinguish individual kinds of cells that may be right next to each other doing completely different things. But with optogenetics, what we developed were ways to introduce genetically encoded light-activated regulators of transmembrane ion conductance that conferred this ability because one can use genetic or anatomical tricks to introduce them into individual cells. And the core bit of biology that made this all work, and I think the photobiology here is one of the most interesting parts of what I'm going to tell you about. The nature of these proteins themselves, their structure-function relationships. But they come from microbial organisms that need to sense light, that do so to live, to survive, to acquire energy. But these are not big cells. They don't have big genomes. They don't have a lot of space to have signal transduction cascades like our retinas do. And so they are very efficient. They use single component light-activated beta regulators of ion flow. And these were initially identified in microbes in 1971. Dieter Osterheld and Walter Stekenius found the first evidence that microbes had rhodopsins, and this was an amazing thing at the time. And they found proton pump activity. 1977, Mukahara and colleagues found chloride pump activity. Peter Hagemann, my Harvey Prize co-laureate, identified cation channel capability beginning in 1985 and leading up to a paper in 2002. And then many more of these have been found since. We and others have found more and more of the members of this microbial opsin family. Now, this has turned out to be absolutely essential to get this optogenetic method to work. There are other strategies had been tried where one would try to recapitulate, for example, the entire vertebrate phototransduction cascade with multiple components, at least 2 or 3 or 4 components, and this turned out to not be practical or tractable in experimental systems to identify new discoveries in biology. But the simplicity and elegance of the single component microbial opsin genes allowed this to happen. So that's why this is such an interesting thing to talk about. They're also just beautiful proteins. How they work is wonderful. And as someone who started as a biochemist and a molecular biologist, I continue to study them for their own sake. But the worlds converged on July 1st, 2004. The editors of Nature Neuroscience asked me to show the notebook page from the very first experiment. And this is what I did on July 1st, 2004. And the notebook page we published in Nature Neuroscience in 2015. But what I did was I had cultured neurons, and I expressed, this was part of a broader strategy in the lab to express many different potential genetic modulators of activity. For example, we had TASC channels and TREC channels. These are potassium channels that I had constitutively active and dominant negative versions of to try to create genetically targeted control of activity that way. But I threw in the mix this Channelrhodopsin gene. It's a microbial opsin. We called it CHOP2 at the time. And I exposed one set of cells to light and another set of cells not. And my readout was, for membrane depolarization, was activation of the calcium-activated transcription factor, CREB, serine-133 phosphorylation of CREB, which I had studied in great detail before, and I knew that its activation was membrane depolarization triggered. And a lot of things were revealed in this experiment. First of all, that the Channelrhodopsin YFP fusion protein was safely produced in neurons, these sensitive neurons, that it was trafficked, even though it was from an algae, that it was trafficked reasonably well, went to the membrane, even down processes somewhat, but also that the illuminated cells had activation in the nucleus of serine-133 phosphorylated CREB, and the non-illuminated ones did not. And it's a very humble experiment. I just kept a simple tally of which cells were activated and which were not, and I always like showing this. This whole experiment probably cost, in total, probably $100 or so. But it was what showed that it was going to be possible to use these algal genes in neurons to activate them. And this went on over the next year. I had a very small lab at the time. This group worked very hard over the next year in 2004 to 2005, leading to a small paper in Nature Neuroscience in 2005. And this is the small group here, the 2 students, Feng and Ed. Feng Zhang, now famous for CRISPR genome editing work too, but back then he was just a chemistry grad student in my lab. But you could tell he was brilliant even then. And we did a lot over that year. Electrophysiology showing you could flash brief pulses of light and drive action potentials in neurons, building viral vectors to allow us to deliver and get stable long-term expression of the microbial opsins in neurons. But we got a lot of criticism. This was a small paper in 2005. It was purely in culture, and a lot of people said, and very rightly so, well, that's a nice trick in culture, but you're never going to get this to work in behaving mammals. You're not going to get high enough expression that can be targeted in a versatile and general way. How are you going to get the high-intensity light in? Where's the chromophore going to come from? All transretinal, which is the small vitamin A-related molecule that the microbial opsin folds around, and it actually receives the photon. And all this was true, but we soldiered ahead anyway. Feng, this is his early sketch from 2006 of a design for a fiber optic device, and this is actually how it pretty much looks like now. So he was a good artist as well as a great scientist. But this was the moment we solved a lot of these problems over the next 5 years. In 2007, Feng did this experiment that really revealed that it was likely going to work. This is a mouse that we made together with George Augustine and Guoping Feng, and what this mouse does is express Channelrhodopsin 2 in layer 5 pyramidal neurons in cortex. And Feng implanted a fiber optic in right supplementary M2 motor cortex with the hope that when turning on the light with the fiber optic interface that he'd see some sort of movement toward or attention to the left side of the world. And you can see when the light goes on what he saw. So this was the moment when we really knew that it was going to work, and this, you can appreciate how it felt to see this. And then as soon as he turns off the light, the animal stops and is completely fine. And so this, by itself, there was no scientific finding there, of course, but it was the start of many things. And so we went on rapidly from there to target hypocretin neurons using a promoter fragment that we could fit into a lentivirus, about actually quite a small promoter fragment, less than 3 kilobases, and we could fit it into a lentivirus along with Channelrhodopsin. And this allowed us to target hypocretin neurons in the lateral hypothalamus, and in a collaboration with Luis de la Sella, we were able to show that certain patterns of spike firing in these deep neurons triggered the state transition of sleep to wake, but other patterns didn't. And that was in 2007. Now, that is the very brief background of getting to something that worked in behaving mammals. Before I come back to applications, including into the zebrafish, which is some pretty exciting unpublished work, I want to talk a little bit about the proteins themselves, just because they're beautiful and we care a lot about them, and a lot of active work on those proteins has allowed new possibilities for neuroscience. So what are they? What are these proteins? Well, they are 7 transmembrane proteins. They, as I mentioned earlier, fold around an all-trans retinal. The proton pumps and the Gaillardiopsins, the chloride pumps, the crystal structures have been solved over the ensuing decades, and a great deal of wonderful work was done on the photocycle. For example, the pumps carry out a discontinuous series of ion handoffs. The ion coming in one side of the protein is not the same as the ion that goes out the other side. And there's a beautiful literature, thousands of papers over 34 years on photocycle analysis of these pumps. However, the channels were a complete mystery. It was clearly, there was clear sequence homology. There were 7 transmembrane proteins, but there was not a precedent for a channel to be formed by a 7 transmembrane protein, much less a light-gated ion channel. And there was a lot of debate and controversy. First of all, was it really a channel? Secondly, if there was a channel, could it be within a 7 transmembrane protein? In fact, the leading theory was no, that it would have to be formed at the interface of 2 monomers, 2 7 transmembrane proteins, and that was even described in a paper from a low-resolution cryo-EM structure. But getting from this level of understanding to a detailed structure and structure function analysis has been really powerful. We can now do amazing things, play in and use very diverse and versatile kinds of transduction logic, photon spike transduction logic, not just using blue light to drive action potentials, but we can use red light at high rates. We can have bistable excitation, so flip cells into and out of excitable states that are stable and that last exactly as long as we want, or bistable inhibition, flipping cells into and out of inhibited states as well. So a great diversity of types of logic we now have access to, and each of these advances arose from either structural models or crystal structure plus additional engineering. A great deal of this work was done in collaboration with Peter Hegemann, the co-laureate. And I'll tell you a little bit about some of these very briefly. For example, we can speed up the spike firing greatly if we want to. There were some issues early on. The deactivation of Channelrhodopsin wild type is about 10 milliseconds. But that's pretty good for a plant. It's pretty slow for a fast spiking neuron. And we noticed some problems. For example, occasionally we'd get doublets of spikes instead of a single spike. And that was due to the Channelrhodopsin not turning off quickly enough after the light off. But we found ways mutating this glutamate 123 to threonine, or out, threonine being the best one to speed things up effectively. We also, I mentioned earlier, this bistability. We could slow things down greatly too. Here's that very fast deactivation after light off, 10 milliseconds or so in wild type. But we can greatly slow that deactivation by many orders of magnitude, even now tens of minutes before it'll spontaneously turn off. And we retain control over the off because we can use a red-shifted photon to turn it off. So blue to turn on, stop the light, but it stays active, and then we can use a red-shifted yellow, green, or red photon to turn it off. So we still have high temporal control, but it's bistable. And these are amazing for many purposes. I'll tell you about them later, but they allow you, first of all, to not have to keep dumping light into the system. Secondly, the cells become orders of magnitude more sensitive to light because they can integrate photons. Because the opsins don't turn off, very weak light can, if delivered for longer, can elevate the cell up to its maximal photocurrent level with extraordinarily weak light. And so this gives you access to very large volumes of tissue. These two advances were done strictly by structural modeling. So this was done with mutating this cysteine 128 and this aspartate 156 based on homology modeling with the pumps. But this only took us so far. For example, we couldn't get to pore re-engineering because we didn't even know where the pore was, even if there was really a pore. And so we really wanted access to that. We wanted to change ion selectivity, for example. But the pumps had no pore, and we didn't know where the pore would be. So we worked very hard on this in collaboration with Osamu Nareki and Hideaki Kato and Feng Zhang and Ofer Yizar also, who is one of my postdocs now at the Weizmann. And in 2012, we got to the crystal structure of Channelrhodopsin. And this is what it looks like. It is indeed a dimer of two 7-transmembrane proteins, but there's no pore at the interface. We can see that clearly. There's no charge or polar residues lining this interface. But each monomer clearly has its own pore. Helices are tilted farther back than they would be in another 7-transmembrane protein that creates a pore, and we can see it's lined with charged and polar residues. This magenta is the all-trans retinal. So this led to a question, with this knowledge, can we now create new ion selectivity? The wild-type ones at the time were all cation channels, non-selective cation channels that anybody knew about. So they fluxed sodium, potassium, calcium, and protons, but did not flux anions at all. Now, the interesting thing, looking at the pore, it was, there were no bound ions, there were no high-affinity binding sites that we could see. It was a bit of a big pore, a bit of a disordered pore, but one theme was clear, that it was lined chiefly with negative residues. We predicted, calculated a negative surface electrostatic potential in the lining of the pore all the way through. And we thought, okay, well, if this is the reason it's a cation channel, maybe we can change that. And so Andre Barrington and Sue Lee in the lab carried out a heroic series of experiments trying to completely reupholster the whole inner lining of the channel from this largely negative to a largely positive environment with the goal of creating anion selectivity. And you can imagine, how many things could and did go wrong with that. You know, all this, of course, the pores there, but the retinal is right there too, and you can imagine, as was certainly the case, many of the mutants didn't work or had altered gating and so on. But we looked at hundreds of different mutants and put them together over the years, and indeed they did shift the lining from largely negative as shown in red to largely positive as shown in blue. Hundreds of different mutations pieced together. And in the end, it was indeed a chloride Channelrhodopsin, and we could turn off spiking with blue light instead of driving spiking. And so this was exciting for a lot of reasons. We described this in 2014, back-to-back with a very nice paper from Peter Hegemann, different mutations, same result. This was great because channels are more efficient than pumps. They can move hundreds of ions per photon instead of a single ion. But also, there's no way to make a bistable pump. It just moves one ion and that's it. You can't stick it in an open state like you can with a channel. But now we had an inhibitory channel, a chloride channel. Chloride flux, under most circumstances, is going to be inhibitory given the typical ion balances seen in physiological systems. And so we said, hey, this is great. Maybe we can make a bistable inhibitor for the first time. And that worked. We combined these mutations with our earlier bistable or step function mutations, and this is how we got this sort of logic of bistable inhibition. 10 mutations in the pore, and then the step function mutation. So this was exciting, and then a very interesting thing happened in 2015, a year later. So we did this in 2014. We were working on applying and advancing the use of this new set of channels. And then John Sputich's group in Texas found and described in 2015 a naturally occurring chloride-conducting Channelrhodopsin, which was amazing. I don't know why. Maybe people could come up with some theories why this particular organism wants a fast, light-activated chloride channel. It's an interesting organism, Gaillardia theta. It's a microbe. It has a lot of the same ecology as Chlamydomonas reinhardtii, where the original Channelrhodopsin we worked with came from. Anyway, it does, and it's a naturally occurring chloride-conducting Channelrhodopsin. So we were very excited when we saw this, and we said, okay, let's go look at the sequence and see what the predicted pore electrostatics would be and compare them to what we had generated a year earlier. And they're almost identical, which was really amazing. So this is the Gaillardia theta replacements on our Channelrhodopsin crystal structure. And this is what we had made and published a year earlier. So this was really thrilling because it suggested that we understand the pore selectivity mechanisms. Of course, nature always gets there first. I'd like to know more about the behavioral ecology of this organism, but it was an amazing thing to see. So that was exciting. And then, but that's only the tip of the iceberg. There are organisms that make spectrally shifted opsins as well. Feng Zhang, again in 2008, had found from Volvox carteri, this multicellular green algae. This was, again, together with Leif Fenno, Ofer Yuzar, and Peter Hegemann, a Volvox Channelrhodopsin VCHR1 that we then engineered over a number of additional years to make a very high performance red light activated Channelrhodopsin called C1V1. Now, this was described finally in 2011 in a collaboration, the same group of people plus Matthias Prigger, who's worked with Ofer here in Israel as well. Now, spectral properties are a little harder to predict than the other properties. The kinetics, faster, slower, the pore selectivity, I mean, those were exciting and thrilling at the time, but we and others had a great deal of difficulty meaningfully shifting the action spectrum of the opsins. We wanted to redshift them as much as we could for a number of reasons. One is that we wanted to get combinatorial control. We wanted to have two independent channels of communication with two colors at least. But the action spectra are broad, and even with engineering, even with screening, nobody was really able to shift them by more than 10 or 15 nanometers, which, given the broad action spectra, was not very useful. There were theories about this. So, you know, In 2005, Klopman et al., for example, described a theory, again, based on electrostatics around the all-transretinal polyene that has to do with the energy needed to drive electrons up a electrostatic gradient environment, and lure higher energy photons being needed to drive up a steeper gradient. But this didn't translate directly into being able to engineer the spectra as we wanted. There are other factors that seem to control spectra, long-range interactions across the protein, the planarity of the polyene, and other factors. So very difficult to engineer. But the VCHR1, the Volvox naturally occurring one, was already, nature had done it already over millions of years of evolution, and we were able to use our tricks to shift it a little further, and we ended up with creating a C1V1, which can be driven nicely in 600 nanometers or above, and you can drive spiking, as we showed in 2011. Now, C1V1, it's called that because it has multiple mutations. It's also a chimera between Channelrhodopsin 1 and Volvox Channelrhodopsin 1, so C1V1. It can be used for combinatorial control, but the biggest advantage of it, we didn't even appreciate at the time. It turned out that it's very well driven by two-photon illumination and is nicely compatible with a separate stream of work coming from other labs, which is development of genetically encoded calcium indicators, the best ones of which are blue light excited. So the two-photon point, Ofer and Rohit Prakash in the lab showed this in 2012. People had a lot of trouble driving Channelrhodopsins with two-photon illumination, but C1V1 seemed uniquely suited, and this was the first in vivo living mouse demonstration of that. So patching a loose patch with a pipette filled with a red dye in neocortex of a living mouse, and then scanning two-photon illumination just above the cell, not getting a spike, within the cell getting spikes, and then just below the cell not getting a spike. That's in Z, the most difficult direction to get this effect due to scattering and optics, and it was even better in the XY direction. And so this turned out to be useful. That was an electrical readout, but now with genetically encoded calcium indicators, you can have all optical experiments. And so, for example, in 2014, together with David Tank, we showed in hippocampus that you can do all optical single cell resolution experiments, play in and read out with light. And this, many labs are now doing all over the world, including Shai Shoham's group here and Michael Heuser's group, Rafa Yusta's lab, Hillel Adesnik, and many others. And also ourselves, we're continuing that as well. This is work from Jim Marshall in my lab in visual cortex, hopping from cell to cell with a point of light, pinging different cells. These are cells co-expressing a genetically encoded calcium indicator, GCAMP, driven by blue light, and using C1V1 to excite cells. And you can do this with two-photon or with red light, pinging individual cells, seeing how they respond. Cell numbers indexed here on the y-axis and time on the x-axis. And this color scale shows intensity of the calcium response. So that's single cell resolution work. You can also, in many cases, back off from single cell resolution. Since you're using genetically targeted tools, you can leverage the fact that you know where the photons are coming from and which cells are being excited, and just collect photons rather than forming an image. And this turns out to be very useful. We described this in 2014, fiber photometry, and we set it up to look exactly from the animal's point of view, just like the optogenetics experiment. So same fiber, same placement, same diameter, chronic fiber, everything the same from the animal's point of view. And that was by design so that we could do all optical experiments in the same animal addressing the same population. What makes this work, so for example, you can target deep brain dopamine neurons in the ventral tegmental area, and using some of the recombinase dependent viruses that Feng had designed in 2009. But targeting dopamine neurons, and then you have an animal in a freely moving animal carrying out a rewarding task, sucrose consumption. Mice like sugar or water just like we do. and that's a rewarding, motivating thing for them. But if you just express yellow fluorescent protein and the animal licks for sugar water, as each lick shown in the red dash here, you don't see a response. That's just because there's yellow fluorescent protein, but not an indicator. If you express a genetically encoded calcium indicator, you can see it with every bout of licking, these little blips of activity in the dopamine neurons. And this is in a brightly lit lab room, deep targeted population of neurons. And so you're collecting in real time the activity of the targeted population, not forming an image. So it's photometry, not imaging, but you still know exactly which cells the signal's coming from. And the reason this works is we have basically a spinning pinwheel, an optical chopper that breaks up the light in 400 hertz. You can set it at, for example, and then on the outgoing direction, you have a lock-in amplifier designed to focus on light that's modulated at that frequency, which nothing else in the room is modulated at. And this single time-varying scalar that you get out of photometry is really useful because it's something that lends itself well to rapid analysis and thus closed-loop systems. This is not published yet, but Noah Young and Tina Kim have now taken us from the way most optogenetics experiments had been done historically, open loop, maybe guided by what you know from the literature. We think these cells are spiking at 10 hertz or 20 hertz or 1 hertz. Playing that in, activity guided, but still open loop, to being more like a well-engineered system like your car's cruise control, where the system readout then feeds back and guides what's being played into the system. And this now works with photometry quite well. You can play in and read out in real-time closed loop. This is a demonstration of that. Dopamine neurons co-expressing C1V1 and GCAMP or a variant thereof. And here's this single time varying dopamine neuron scalar. They collect mean activity in the gray period, and then they let the system free run. This is what's happening as the animal is behaving, and then they turn on the closed loop software right here to fix it at this dotted line, and it does a pretty good job. So you can now do all optical closed loop work. And this can be used to match what's naturally occurring in that same animal. And this is something we wanted to do for a very long time in optogenetics. And it may seem simple, but I think you appreciate it. Tina Kim in 2016 showed how this works, targeting deep brain dopamine neurons. This is a C1V1 variant called breaches for red light control, GCAMP for blue light readout. And this light blue trace is the animal's natural dopamine response to sugar water. So that's the naturally occurring population response. And then she tried to provide that optically. Came in too strong, clearly first, but then she could just back off in light intensity until she hit it, the naturally occurring timing and magnitude of the dopamine neurons in that same animal. And so that, of course, is something that we really like to be able to do. You can also do activity-guided and closed-loop work at the single cell level. We and others are using spatial light modulators here targeted to these two red square boxed neurons. When the red asterisk comes on, that's when we're delivering stimulation to these two cells that we had picked out because of their naturally occurring activity pattern in visual cortex, a particular direction selectivity. So you can do that as well. And so this is what you might call sort of modern optogenetics. It's, you can work on any time scale, acute or chronic, excite or inhibit on any time scale. You can have multiple fibers across the brain, as Tina showed in this 2016 Nature Methods paper. Target cells, cell types, projection types, all in behaving mammals. But crucially, you can now match or modify naturally occurring activity patterns in those very same circuit elements during behavior. So this now lends some color to that photon spike transduction logic, and I think you appreciate why we work so hard to understand the proteins themselves, engineer them, so we had the capability to do these sorts of experiments, and we have an understanding of now why all these things work and how they advance the neuroscience, but also why they matter to the protein itself. So now I want to talk about some unpublished, very recent work with application to zebrafish and to complex behavior. And it was mentioned I'm a psychiatrist. I focus on patients clinically who have depression, that treatment-resistant depression. That's my clinical specialty. A lot of these patients are very anxious as well, and I'm not working in the lab on any sort of direct clinical translation, but we are inspired by clinical questions. We use ideas and insights from the clinic to guide us in our work. A lot of our work on anxiety uses this sort of task. This is the so-called elevated plus maze. There's a closed arm that the animal chooses naturally to spend almost all its time in, very rarely venturing out into the open arms, which are very exposed. It doesn't have these elevated walls. And what Kay Tai in the lab had found in 2011 was that, and when the blue letters come on here, that's when she started playing in control over a particular pathway in the amygdala that turned out to be anxiolytic, anti-anxiety. She's activating this pathway right now, optically, and for the first time, the animal's willing to go out into the open arms of the maze. And it's not changing anything else about its behavior. It's not speeding up or slowing down or becoming risk-seeking. Still spends time in the closed arms as well, but it's willing to go out in the open arms for the first time. And this was a very reliable result, and it reversed when we turned the light off. And this, identifying the causal impact of different circuit elements, has been really powerful. And that's just one feature of the anxious state, which is risk avoidance. We found that a projection from the bed nucleus of the stria terminalis to the lateral hypothalamus was causally involved in this risk avoidance behavior. But anybody who's been anxious knows that's not all of anxiety. Anxiety also has physiological changes, heart rate, respiratory rate changes. We found that in later work with Seung-Hun Kim and Avishek Adhikari, that was due to a projection from the BNST to the parabrachial nucleus in the brainstem. That's where the respiratory rate changes came from. Anxiety also has another feature that's very important, which is that it feels bad. And this may seem obvious, but it's not. If you had perfectly good risk avoidance, why do you also need to feel bad for anxiety? It seems like a cruel thing for nature to do, but it certainly does feel bad. That's why it's clinically significant. And what was very interesting is we found this sort of intervention where you have a pure risk avoidance effect, the animal doesn't care that's happening. If you have it in a separate situation where you test its preference for being in one place or another, and you drive that exact same pathway, the animal doesn't care. It has no subjective valence that this pathway is being driven that causes risk avoidance in the elevated plus maze. Doesn't, as far as we can tell, the animal won't report to us that it feels good or bad to the animal, if that's a phraseology I can use. It certainly doesn't reflect that in terms of its conditioning. That instead is controlled by a separate pathway from the BNST to the ventral tegmental area. And all these different features of anxiety projections come out from the BNST and recruit them across the brain, and they get assembled into a coherent state. There's local excitatory recurrence in the BNST that we think could play a role in making the state coherent. So this sort of insight couldn't get without the circuit element specific control. That's anxiety. What I want to tell you about now is something that's more having to do with behavioral state transitions that we see in depression. Now, we don't work in animal models of depression. We don't claim animals are depressed. As I said, we just take inspiration from the clinic. And one thing that we see happen in people with depression is an extreme version of a natural, adaptive behavioral state transition, which is from active coping to passive coping. We've all experienced this transition when there's a challenge or an environmental situation that is too difficult. One transitions with some individual variability or experience-dependent variability from actively coping with the challenge to a more passive approach. And this is a natural and important thing for animals to do, but people who have depression clearly transition much earlier and much more quickly and much more stably to the passive coping state. It's a hopeless-like state. And animals, not even disease models, but rodents will do this perfectly well. You can put them into a challenging situation, a cylinder filled with water, and they'll try to get out for a while, and then they'll realize they can't, and they transition from the active coping state to the passive coping state, and they simply float. Melissa Warden in my lab did a lot of work on this to understand the causal circuit elements that are involved. She set up fiber optics and fast real-time collection of individual kicks the animal made, picking them up with an induction coil. And you can actually see that transition, that behavioral state transition happen before your eyes here in these data. The kicking active, coping, and then transitioning to passive. Now, a lot of people have studied this over the years in various forms. There are many different types of model system that this has been studied in. Many individual regions have been implicated in various ways. Melissa Worden found in 2012 optogenetically that there was a projection from medial prefrontal cortex to the lateral habenula, a deep brain structure, that when she excited this structure, she favored the passive coping state. And you can do this sort of experiment. You inject a virus here, but then you illuminate here. The opsins, particularly if you help them with some engineering, they traffic very well down axons, and the only light-sensitive element are the prefrontal neurons that project to lateral habenula. And she saw this small but very consistent, reversible, and repeatable effect. Every time she illuminated, the animals became reversibly more passive. And this was not seen if you just had yellow fluorescent protein. She was expressing Channelrhodopsin. And it was not a non-specific motor effect. It didn't affect their movement in the open field in a non-challenging situation. And the anatomy mattered a lot. The prefrontal projection to the amygdala had no effect, and the prefrontal projection to the dorsal raphe nucleus, where most of the serotonin neurons in the forebrain live, had the opposite effect. This favored active coping. Every time she drove that projection, she saw active coping. And again, that was specific, not a non-specific effect. So very interesting individual circuit elements coming from prefrontal cortex, where we think a lot of advanced motor planning comes from, some favoring active coping, some favoring passive coping. Now, this was interesting, but it was very, as you can see, very guided. You have to think about where you're going to inject, where you're going to implant, and it's not like you're doing a Brainwide screen. And so we were, it's like that parable of the guy looking for his keys, but only looking under the streetlight because that's where he can see. That's kind of what we were doing here, kind of guided by very specific ideas. And so what I want to tell you about now is some work that's completely unbiased, brain-wide, single-cell resolution, active to passive coping state transitions. And we do it in fish. So this is not yet published. It's something we've been working on for many, many years. These are our tanks. I like my tanks, but I won't dwell on the tanks. Instead, let's talk about zebrafish anatomy. So they don't really have a cortex. They do have all the same neuromodulators. They're the same basic vertebrate brain plan. They have a lateral habenula, medial habenula. They have a raphe. They have VTA, dopamine neurons. They have a lot of the same basic structure. The bigger problem was, is it possible to study a behavioral state transition like this in the fish? Well, you can't do a forced swim test because they like swimming already. So Aaron Andelman and Vanessa Burns worked very hard on this, and what I had tasked them with was to find a behavioral state transition, active to passive, in fish that were young enough and small enough that we could still see optically every single neuron in their brain. And they did it finally. And what they did is shown here. So they're using free swimming, day 10 to 14 post-fertilization zebrafish, free swimming in a tank, and they give a mild 1 hertz electrical shock, which the animal cannot escape from. So it's an inescapable stressor. Normally, first half and second half of some 20, 30-minute behavioral epoch, the fish swim equally well, as shown in the purple and orange. But if the second half of the epoch is with this inescapable shock, you can see the animals enter into a passive state. Now, a lot of controls, you may want to ask, are you just damaging the fish in some way or searing them lightly or something like that? But they did a lot of controls and over the years and turned out to actually have the properties that we were interested in. So you can see it here. First, there's this active coping state, and then they transition to a passive coping state over time. Very interesting kinetics, not too dissimilar from the mammalian case. They recover fully. As you can see in this trace, they recover back to control levels if you put them in a new tank with new water. Not instantaneously, not as you would see in a contextual fear conditioning case where you put a mouse out of the dangerous context in the new context, it immediately starts moving around normally. takes some tens of minutes to recover, but they don't recover if you keep them in the same tank or keep the same water. So it's a context-dependent passivity, okay? And not only was that true, but also it was pharmacologically blockable. This is the normal duration of our test, and with the fast-acting antidepressant ketamine, as shown here, we could greatly slow and diminish the transition to the passive coping state. And this is animals that were still moving normally, so they were not anesthetized and still responsive to pain. So then we had this behavioral state transition. Active to passive coping had a lot of the properties that we wanted. Question, of course, what imaging modality to use. We've worked very hard as anybody over the past few years on designing optical strategies for getting single cell resolution, volumetric readouts of activity. Things I won't spend a lot of time on, light field and light sheet methods. And also other people, of course, have worked on these as well. There's some trade-offs. There's a resolution speed trade-off, light field being fastest and worst resolution, 2-photon being slowest and best resolution. We worked hard on the light field back, publishing our first paper in 2013 on this, and it's pretty good. This is light field in the fish, and you certainly have single cell resolution where you have sparse labeling, but you can appreciate this here. That in dense clusters of cells, you can't necessarily pick out individual cells. And what my goal, and I'm glad that we stuck to this, was to use a method only where we had absolute certainty of single cell resolution. Still, the light field was useful in some contexts, and so we had a really interesting finding with it. So we took fish, collected baseline activity, genetically encoded calcium indicator expressed in every neuron, nuclear localized for resolution, did the behavioral challenge protocol, and then put them in the light field scope just to image, see what was different. And one brain-wide, so totally unbiased, not just looking under the streetlight, and one region had elevated activity, and that was the lateral habenula in the fish. And so this is the lateral habenula here, and that effect was blocked by ketamine. So this was amazing that with a totally unbiased approach, and this gave us a lot of confidence also in the model. There was some validity in the correspondence to the mammalian case. But we couldn't quite see every single cell. This was a habenula-wide measure. So we went back to good old 2-photon, worked on ways to speed it up as much as we could. We have an animal that's head fixed in agarose. Its tail is free, so we can see it wiggling its tail, and we can get active or passive information from that. And we can collect many tens of thousands of neurons at 1 to 2 volumes per second, which is not much slower than the indicator itself, so we're going as fast as the system really needs to be. And this is the sort of data you get, and you can see true, even in dense clusters, single cell resolution. And actually, this is an animal going through the inescapable stressor, and it's going through the behavioral state transition. And you can start to see something happening here. The movie's going to loop around, but these two lateral structures here, those are the lateral habenula structures. And you can see activity accumulating in those two lateral structures as time goes on. Still dynamic, and now it's looping back, and so it's starting from the beginning again. You'll see this happen again. It's not as if the cells are dying or being pegged to some high stable level. The individual cells remain dynamic, but enter into higher probability activity states. And it really is, the elevation is really just in the lateral habenula. And we can quantify this quite well, looking all across the brain. Aaron Endelman wrote some very nice software to segment every neuron in the brain, and we can see the lateral habenula activity ramping up over time. It's the only structure that does this. There is a very interesting decrease that's seen, and that's in the raphe. And so this is also very interesting. You remember the opposite direction effects of prefrontal to habenula versus raphe in the rat. Raphe goes down, in this case, just a little bit, whereas the lateral habenula goes up. A lot of other things, even the telencephalon, don't change that much. The dorsal thalamus also had a little decrease. Now, that's just a correlation. We don't know that that's causal in anything, but here's now where optogenetics comes in and provides something useful. So what if you drove the lateral habenula? What would happen? And here we can use zebrafish genetics. There are drivers that allow you to express just in lateral habenula. As you can see, we get good expression of Channelrhodopsin in lateral habenula. Talia Lerner in the lab did some zebrafish patch clamping to verify that we were getting the excitatory responses that we were hoping to see. And so what happens behaviorally? Well, it's very interesting. So if you take naive fish that have not been through stressor, inescapable stressor, and also fish that are not expressing Channelrhodopsin, as shown in black, if you expose to blue light, there's a little elevated activity that's reversible. So blue light makes fish swim faster. Fish that express Channelrhodopsin in the lateral habenula don't do that. In fact, there's a slight decrease, and this difference here is significant. Small effect, but interesting. Fish that have been through the prior experience of the inescapable stressor show a profound response. Again, the Channelrhodopsin negative fish just show this slight reversible increase, but the previously inescapable stress-exposed fish have a profound decrease in activity in response to driving the habenula that's actually stable. So it does seem as though the lateral habenula can drive passivity. Also now we have the amazing opportunity, we can image across the brain too while we're driving the lateral habenula to see what this state is. So now we can not just behaviorally induce the state, but we can, with a precise optogenic drive in a complementary way, see what happens, how the state is manifested with single cell resolution across the brain. And so here we're using GCAMP6 for readout and Channelrhodopsin C1V1 for control. And we can play this activity. We can even guide light. We can do this in the head fixed case. We can guide light to the left or right habenula. And here's what you see. First, in the first couple seconds, only one, when you're driving one habenula, that's also the only structure in the brain where we see a response. It's good that there is a response there. That means we're doing what we think we're doing. But not a lot that we can pick up across the brain in those first couple seconds. But then 8 to 10 seconds later, we see a decrease in the raphe. So we think the habenula not only can be causal in driving passivity, but it can be causal in driving this raphe decrease. Now, this isn't even the most interesting thing, which I'll show you next. Nothing that I've shown you yet has really leveraged the single cell resolution. But I'm glad we stuck to that because that was, I think, where the most amazing result was. And here's the same movie I showed you before, but what you can see, maybe you can appreciate, it's not as if all the cells are coming up together in activity. And it's very clear that some are late transitioners, some are early transitioners, some single cells, and some are in the middle. And we were sort of intrigued by this, and so we looked across all the neurons, and first of all to see is there any organization within the habenula, is any sort of spatial localization, so just color coding early to late transitioners. And there's not a big correspondence. There is a statistical clustering of early responders. You can pick up that a little bit in this color-coded max projection, but it wasn't a big effect, and we don't really know yet what to make of it. But what is clear, and this is uniquely in lateral habenula, there's a complete temporal tiling across the entire duration of the stressor. Individual cells tick on at different times, but you're covering the whole space. So it looks as if the habenula is set up to measure a statistic about the nature of the stressor, its duration, and it's doing that by the number of cells that get recruited over time. At the end of our period that we've been using, still only about 20% of the cells in the habenula have been recruited. The raphe doesn't do that. The changes that we see there all occur in all the cells roughly about the same time. So it's a habenula property. And I'll close this part of it with just showing you all the data streams that we have together. So this is a control fish, and you can see activity on the left. This is the raw data, and then this is the segmented and color-coded data. This is the actual fish movie. You can see the fish here in that little movie in the lower left. You can see each little tail flip that's made, and that's read out by the little ticks. That's just a control head-fixed fish, and what I'll play here, this is the fish experiencing the inescapable shock, and you can see The shocks are shown in the gray ticks, and you can see happening right before your eyes this behavioral state transition. The animal stops having the tail twitch, and then you can see the activity accumulating in the lateral habenula, both in the raw data and in the segmented data on the right. So I think you can appreciate, I mean, we're just in the very earliest stages of being able to deal with these amazing data streams. We can, you know, what we've shown so far, what I've shared with you is what we know now, but there easily could be, and I'm sure is, much greater complexity in these data streams that we haven't yet appreciated. One big question, of course, being who are these individual cells, the early, late transitioners? How are they wired up? Do they have different molecular phenotypes of one kind or another? And you know, we and others are doing a lot of work to register activity patterns during life with individual cell molecular properties, and so we can register live and fixed GCamp, which normally don't overlap very well, as shown in red and green here, due to size changes that happen during fixation. But we can morph the two data sets together and get single cell resolution, so then we can do staining and use our clarity-based methods to get wiring information from those same cells that we knew had particular activity patterns during behavior. In the last 5 minutes or so, I'll just briefly describe this final data stream, which is integrating with the wiring. So in 2013, we described a method for making intact biological tissues transparent. We do this by removing lipids. There's, as we showed, this allows transparency to happen due to a reduction in light scattering. And the way we make this non-destructive is we build a hydrogel in the tissue before we remove the lipids that locks all the amine-containing biomolecules, proteins, and nucleic acids in place where they remain for further analysis. So you're transitioning the biomolecules to an anchor that's a gel. The gel, of course, as we showed, is stable. It tends to expand substantially during this process. But at that, you can also shrink that back down, as we showed in 2014. You can shrink it back down using at the refractive index matching step. So you can have very rich molecular interrogation. You can do not just protein labeling, but as we showed, you can anchor RNAs as well. In March of 2016, Emily Silvestrok showed this, and you can get very rich molecular information, activity information as well, immediate early genes, and so on, using the RNA labeling strategy. And now, I won't dwell on this. We've reviewed a lot of the different variants now that have come out. There's a whole alphabet soup of different acronyms that we and others have described, different variants on this hydrogel process. Imaging is limiting, or had been limiting. We and others have been working hard on light sheet methods to collect rapidly, still high resolution clarity volumes, and then analyzing becomes a big challenge as well. But we've automated that as well, at least in the mammalian case. We can do whole brain, many animal cohort scale analyses, and we can do immediate early gene, brain-wide type assays, counting all the cells, automatically segmenting them and counting them across the brain, and still retaining wiring information. So here what's being shown is a cell in prefrontal cortex that projects all the way to the nucleus accumbens. The same individual cell is shown in red. We can see it's an NPAS4 expressing cell. That's a particular molecular marker that we found was interesting. But we can see the very same cell that we know projects to the nucleus accumbens is an NPAS4 expressing cell in cortex. And Lee, and Will Allen in the lab had shown that this was an interesting population. It gets recruited by positive but not negative experiences, the PFC NPAS4 expressing cells that project to accumbens. Very interestingly, there's the projection to the lateral habenula is more recruited by negative experiences, and I think you can appreciate how interesting that is. And the projections to VTA are equally recruited by both positive and negative experience. So this is, the hydrogel tissue hybrid method has a lot of interesting properties because it's aqueous-based. It doesn't quench endogenous fluorophores. You can do multi-round labeling, wash on, off, elute antibodies or nucleic acids. You have this reversible size change property, and you can look at proteins and nucleic acids. And now we're just working on integrating all these these data streams together in behaviors of interest. There's a lot that's difficult about this, but it's pretty exciting too, and it's always, you know, amazing to look back and appreciate the microorganisms that made it possible. So that's what I wanted to share with you. I think I've mentioned all the key people along the way, but I want to just thank them again for all they've done. Of course, they're an amazing group of people. If any of you come and visit, come see us in California. And I want to thank our collaborators, and I'm really honored to be here and tell you about our work. So thank you.
Is there a way to go the other way around, to have the neurons that are activated to meet, like chemiluminescence or something?
Yeah. We've actually worked on using bioluminescence sources from neurons to try to drive the microbial opsins, if that's one thing you're thinking about. But you also might be thinking, can you run the microbial opsins themselves in reverse and get a readout of voltage, because they're in the membrane? Can you get a readout of voltage from the opsins themselves? And you actually can. Adam Cohen and his group at Harvard have shown that. So what we have found, though, is the, going back to the bioluminescent question, is the light power densities that are generated are not strong enough, and you give up a lot of temporal resolution with that strategy too. So we tend not to use it. It's a good thought.
And can I follow up? Do you know the molecular targets? Can you follow the molecular targets at the protein level of specific activation in specific neurons, see if they are different, you know, such as post-translation modifications and things like that?
Yeah, we do a lot of so we typically often are very interested in physical properties of the cells that we have controlled, and many things we care about in those cells. First of all, we want to know what their native wiring and structure is. We want to know what they naturally express, what their molecular profile is, transcriptome, and so on. But then we also want to know, what did we do? Did what we do matter for them? And we've looked at immediate early gene expression changes being the most important thing. In terms of structural changes, transcriptional modifications, we haven't done a lot of that. I think that's kind of an untapped opportunity. And it would be pretty interesting. You could see, is there a chromatin or epigenetic signature of having been through one pattern of activity but not another pattern of activity in a particular cell type. That would be pretty interesting. We haven't really done that.
I wonder whether it's possible to induce without the electrode. Can you shine light from other wavelengths that you can go into the brain without using this?
Yeah, without the fiber optic. So the question is, can you deliver light without a fiber optic? And the answer is yes, you can. Actually, back in 2007, Viviana Gradnaro in my lab showed that you could get completely non-invasive through skull control in mammals. She had to thin the skull a little bit with a drill, but it was still an intact skull, and she could just have an LED mounted on the head, and she could drive whisker movements in that case. And that was even with wild-type Channelrhodopsin. Now we have these orders of magnitude more sensitive Channelrhodopsins, and so you can definitely do non-invasive experiments, but almost nobody does, including us, even though we've had the capability for 7 years. And the reason is we really like having the fiber optic is really valuable. That really provides you a targeting capability. You can define cell populations by origin and target very well if you depth target your fiber optic. So we wouldn't give that up even if we could, which we can. Yeah.
About all-trans-retinal, do mammals have sufficient enough all-trans-retinal in the brain to activate these things, or do you have to add it? Or
I'm sorry. Say it one more time.
All-trans-retinal. Do we have it in the brain? Or
Yeah. Yeah. So that I raised that as a problem, but I didn't address it. Yeah, miraculously, and this is something we didn't know would be the case. We were thinking about strategies to infuse all-trans-retinal because it wasn't known to be at the time a part of normal mammalian signal transduction. In the adult case, certainly it's very important in development and so on. Turns out there's plenty of it, and we went back and looked at just experimentally. We found that there's plenty. There's some evidence that you can juice things up a little bit by adding more retinol, but it's not worth the trouble at all, since things work perfectly well without it. We went back and looked at the old literature. There's some old papers in German characterizing retinols from mammalian brains. So people knew about it, but it was in a very old literature that certainly I didn't know about. Yeah, so invertebrates, worms and flies don't, and so you have to add that. But that's easy to do for them. You put it in the food and they just eat it, and that's actually good control.
So, no more questions? Thanks again.
All right, thank you.